thesis defense. Exciting day. Yeah, Zach came to Hilo from Nebraska and actually fell in love with the ocean, freediving, exploring the coast. And he was a standout student in the marine science department, graduated, and then went on and worked in aquaculture industry over in Kona. But stayed heavily involved in a community of spear fishermen and freedivers. And so we would cross paths every now and then, still would see Zach out and about. And then at some point he decided that he wanted to combine that love of the ocean and the freediving with his education and go one step further and pursue his master's. So he came to me inquiring about working with the Mega Lab through the TCBES program. But I was concerned at first because we didn't have a big financial support for the project, and his goals were large. And any of you involved in marine science knows it's time consuming and costly to get a lot of diving work done, especially because his interests were trying to figure out ways, it was really impressively ambitious to try figure out ways that we could improve how we're doing fish surveys, but also bring in people from communities like freedivers and spearfishers and get them involved as well. So I was nervous, and I told him though, hey, look, I think it's possible. Projects like this, where you have strong goals and you have the ambitious drive, they can be extremely successful, but there's a lot of risk of failure, so you've got to want this. And he said he did. And so we gave him the shot and he came on board. And from the beginning, he just blew us away with his effort. Zach didn't shy away from the challenge whatsoever, and he did it on his own. He had to learn a lot of complex statistical analyses to be able to answer the questions he came up with. He had to go out and outside of what you're going to see him present, do an enormous amount of early pilot testing to figure out how the methods would work, how he would access the sites. And so I was really blown away at the effort that he put in. And it culminated in a project that's really impressive, and also was able to leverage a camera system that we have underwater that's streaming. And beyond just his investment in his own methods, he was tied to us installing the second version of that system, which was not easy at all. Involved a lot of diving, a lot of late nights in the dark, torching splices together down on the shoreline at Kona. Zach was the first to volunteer to stay overnight with nothing, to be there till we could get it finished with the insane engineers we work with. Like, he never, ever shied away from the challenge. And he put in all the troubleshooting. And I think he learned a secret that a lot of people don't always realize in science, that it is a long process of troubleshooting. And if you have the work ethic and you're willing to put yourself into it, the success is yours to take. Beyond that, what impresses me about Zach, he never complains. I never saw Zach complain once. He always has a smile. He always comes happy to work. His mentoring ability of the other students that worked with him is phenomenal. He was a true leader, and I'm just so proud to have him be a part of our group. And he's also a fantastic communicator. So it's great to see him go from where he started to where he is now defending. And like I said, I'm proud of all the work he's done. I'm exceptionally impressed with his project, but more than anything at this point, I'm just excited to see where he goes, because he's already been on several Nautilus expeditions. The successes are just layering now because of who he is and the effort that he puts into it. So I want to thank Zach personally for all of his hard work. Thank you all again for coming today. And with that, I'll let him take over and share his work.
Thank you. All right, I got the second best fish shirt in here though today. I think John takes me out on that. But yeah, thank you. I appreciate that. Thank you for the background introduction as well. As you mentioned, yeah, my whole goal was figure out if we are able to enhance our ability to do fish surveys out here in Hawaii. Reason being, like you said, I just was seeing things myself that I was curious of, kind of wanted to put to the test. So I'm excited to get started, and we'll just dive right in. So when we're talking about fish surveys here in Hawaii, sorry, hold on, this is me the other day. Okay, when we're talking about fish here in Hawaii, what are we talking about? We're talking about coral reef fishes. Primarily, these are some of the species you'll see, but mostly there's quite a few families that dominate the reef. So we have a lot of surgeonfish, a lot of butterfly fishes, a little bit of snappers here and there, but really what it is, quite a bit of species. So within those groups, we have a lot. Now with that diversity, I was curious, do we see actually anything between these? Is there anything that's different that we need to notice? Are we grouping these as one when they should really be looked at all separately? So that's kind of what I wanted to do. Now, why are we doing this? Why does it even matter in the first place that we monitor coral reef fishes? They play many key roles. So first off, they are predators and grazers on the reef, right? They're out there keeping the corals healthy. They're out there removing the sick ones off the reef, right? They're out there kind of keeping it intact. As well, they're also excreting nutrients, as you can see from the parrotfish up there, also making sand. They play these critical roles that we need out there on the reef. Third, they're economic pillars. Here in Hawaii, also, they play a role in tourism, right? There's quite a bit of diving. People come here to see these fishes. It is an important aspect. But arguably, most importantly, what they're doing here in Hawaii is they are a food source. People here in Hawaii and also throughout just generally the Pacific area, coastal communities are heavily reliant on coral reef fishes for a food source. In fact, studies have found that estimated $10.3 to $16 million is what you can value these fisheries at. And of that value, majority of it is just subsistent food source, meaning people here eating it, not buying or not catching and selling. They're feeding themselves, their families, and their friends. In fact, if you do the quick math on those numbers, roughly 70% of the value is based on just subsistence activities. So that's pretty substantial. And really, it translates to over 7 million meals annually here. That's a lot of fish, a lot of food, a lot of people. So it just shows the importance they play here and also who's involved. Right now, we have really one method that is kind of the foundation method of monitoring coral reef fishes, and that is using open circuit scuba diving. So as you see, just kind of this is like what you'd see recreational divers wearing as well, same setup. And the open circuit just meaning you are exhaling those bubbles into the environment, right? This method, though, can be quite exclusive. For example, fishermen often are not divers all the time, and so they are not part of these surveys, yet they're very heavily reliant. The freedivers, spear fishermen, people who I was hanging out with, they were never a part of this, but yet they were being told what the rules were based on these other surveys that they didn't really understand. So wanted to see what we could do with that. Before we could put this to the test, though, we had to really figure out how these different scuba methods also worked and what was the best method to use. So there's two methods that are kind of dominate the way people do scuba surveys throughout the world. And it's a stationary point count or the underwater belt transect. The underwater belt transect is the most common one you'll see, but stationary point is right there with it. And arguably, I would say, I might be biased, a better method for it as well. So the way the visual transect works, pretty simple. It's just like you would do it on land as well. You have a 25-meter transect. You swim along that transect, and then you're counting, sizing, and recording all species you see within that 25 by 5-meter boundary or whatever you decide your transect is. So this method can be pretty intense, especially for new divers. It's a lot going on in a short amount of time that they have to constantly be keeping up with. So we'll get there a little bit, talking about one of the issues with that. But overall, this method has kind of been tried and true and been heavily utilized all throughout the world. The second way that people often use it is the stationary point count. So this is the one you're going to hear me referring to mostly today. This is what we use. This is what we compared to also. Essentially what you're doing with stationary point count is you have a diver centered in a cylinder, and they're responsible for surveying a 15-meter diameter cylinder around them. So 7.5 meters in each direction. And then this one's a little different in the fact that the first 5 minutes of your survey typically is where you're recording all species, and then the following 10 minutes you're doing the counting and sizing process. So it's different in the sense that you're not doing everything all at once. So that's kind of the big difference there, other than just the swimming pattern. But that is one thing to note. Now, both these methods, like I said, kind of tried and true at this point. They're accepted everywhere. People are using both of these. However, people have also brought up the inherent biases with these. Oftentimes, people are critical that scuba diving is not the best method for fish because it might be changing their behavior. So the first one that people often bring up is that frequency from the bubbles of the divers is affecting the fish. So essentially that sound of the bubbles is the same frequency these fish are hearing at. So they're concerned that this might be pushing fish away or also bringing some in. So a couple studies have found that there is an effect of that. Building on that, they've also found this is even more important or more obvious in areas with high fishing pressure. And so this means areas kind of where divers or fishermen are oftenly present and kind of predating on all these fish, right? They recognize you as a predator and they're going to become more weary is the idea with that. In areas of less fishing pressure or no fishing pressure, it's likely you would see different results. And then lastly, what I personally think has a big effect on this also is the experience level. And this study found that the experience level heavily influences the counts, especially of specific families. So major families too, things like the Acanthuridae and the Labridae, which really make up a lot of the fish here in Hawaii. They were some of the most problematic fish for the new people to be counting. So when you take all that into effect, there's quite a few things going on there that might be affecting the actual behavior of the fish. It's hard to say specifically it is just scuba diving. There could be other things going on. Another thing to note with these is many of these studies take place over various different locations. They don't do them necessarily at the same spot all the time or the same time of year. They just do them over time. And so when you're not considering all the other variables that are changing, it's really hard to pinpoint the fact that you can say method is the issue and method is changed and method needs to be changed. So this is where this project came up. It was, okay, well, I want to know if this is affecting their behavior, and I want to do it using 3 new ways. The first one is using the remote underwater video. So I'll explain all these much more in depth later, but really remote underwater video, free dive, and snorkel were the 3 techniques we were going to, or that I'm proposing. Now the camera is nothing new. People have used camera for fish surveys. However, you'll see this camera is very novel in the way that it works and also in the functionality it has compared to previous studies with cameras. Free dive. I haven't seen anything in the literature about freedivers. And this is one that really, again, as a spear fisherman, seeing this, it was like, man, these people are in the water all the time. They know more about fish. They're really good at sizing fish. This is a lot of valuable information we're passing up. Why don't we have something that includes these people? So that was a big goal of mine. And snorkel sounds pretty obvious. It's used here and there, but there's nothing heavy in the literature saying this is the best method to do, and it will give you the same results if you did scuba there also. And this one for me also kind of is relevant here in Hawaii and throughout the Pacific because there's many areas that are remote, don't have access to scuba, or it costs a lot of money to bring divers in and survey their locations. So if we can show that snorkel is an effective method, if you're comfortable with snorkel in your mouth and holding a clipboard, you can be an effective surveyor is what we were hoping. So that is the first objective of this. Can we use these other methods? And really what I expect out of this is that the diver-based surveys, we're going to have lower species richness than the camera. And the reason I thought that is because the camera is mounted out there. It is permanent. It's not disturbing. It is part of that environment essentially at this point. Whereas as divers going in, I expected that to result in quite a bit of behavior change and different results. From there, I wanted to look further at this, and I wanted to know how related actually is this or how related are these results, and what is the variance between these methods? So we use 2 methods or 2 statistical methods primarily for this, with the linear mixed effects model and principal component analysis. And from this, what I really expected was we'd be pretty good at counting and sizing when we're in the water. So when we're doing scuba, free dive, snorkel, but I was worried we wouldn't be very good at with the camera. And that was really what we expect, is seeing that camera we can see, but we can't do much more than that. It's going to be a really good kind of more of a social science experiment where we can see what gets people excited about the reef, what are people most invested in, what is it that people are really curious of. So I wanted to really test that out. So to do this, we focused on 1 study site. We were at the exact same location the entire time. So dove this spot. I was joking with Jacob about it. We got in out this spot over 60 times by the end of it. Well, the first time we got in, it was an intense spot. It's the most westward point of the Big Island, known as Keohole Point. So in terms of fishing pressure, it's not necessarily high. There are some divers there, but honestly, the environment there and the lack of ability to get in and out of the water in the area really limits the pressure it has. By the end of it, we felt pretty comfortable. It's good. We still get humbled as we went throughout, but this was the location of the 1 survey. You can see they're kind of in the picture. Let's just zoom in the coastline. It's kind of just lava rock and cliffs right there along that area. So the depth of this survey was 8 to 10 meters for every survey. And the location is where the camera is. So we were positioned on top of the camera for all of these. Now, this quick summary before we actually get into how we did this. The actual final analysis consisted of data from 9 days. The reason of that 9 days, though, is not because that's all we had time for. As John mentioned, we did a lot of pilot data. For over a year, we were driving back and forth, testing these different methods, trying to figure out how can a free diver dive down, remember species, write them down, count in size. How can you do all these things while free diving? And then can you do it snorkeling? And we went through many different versions of each method, and I'll talk more about them when I get to the specific. But there was many more dives than what you just see here of what we actually did. In total, though, in those 9 days, because of all that back work we did and how well we experienced we had been with those methods we came up with, we were able to do 60 surveys in those 9 days we were there. So during those survey days, we had a couple things we wanted to be wary of. The first thing is there are kind of these morning and evening rushes of large schooling herbivore fish, and we didn't want that to come in the middle of our survey and really throw things off. So we set 2 time frames. The morning period was from 8:30 to noon, and within that period, we conducted 1 survey of each method. And then same thing in the afternoon, we did it again. So we'd be there all day long as weather permitted, and we could essentially by the end of the day, we had done 2 of each survey method. The survey order within those time periods, the morning and afternoon, were randomized. So it wasn't necessarily we were doing scuba, then free dive, then snorkel. We completely randomized the order of them. It was always nice when camera was first, gave us a little later start, but not always the case. Again, from there, methods. First one, open circuit scuba using the stationary point count. So this is the reference method. This is everything I'm comparing today. This is what we're comparing it to. Again, this is the tried and true. It's out there. The remote underwater video. Again, this is positioned there. It has been there for years now. Essentially part of the environment there. Free diving, essentially what it is. We're diving down. We're holding our breath and then observing doing that. And then snorkel, just from the surface the entire time. Now, before we actually figured out how we wanted to use each of these methods, first thing we had to figure out was how long do we need to be surveying for? And this is critical, right? And this was a big argument that people had against stationary point count when adapting even here in Hawaii was, how do you know you're actually getting an accurate representation of the entire community? How long do you have to survey for? And they found about 15 minutes was a good amount of time to get majority of it. So we wanted to make sure with these new methods that actually was the same, and we're not just assuming that it's the same. So we went out, this was a big part of the pilot data, and we tested it, and we would sit there and every minute we would just record what new species came in. And you can see there's a pretty consistent trend through this, which means a 15-minute survey for each method was pretty equal across the board. So what those actual surveys ended up looking like was following kind of the tried and true method. So the first 5 minutes was species identification. So just you do an entire rotation, record every single species you see. The second 10 minutes, 5 to 15, that was counting and sizing. So you kind of systematically go through your list of species you have, and then you're counting and sizing them that way. Now, the open circuit scuba, again, there's not much here to talk about other than this is the reference method. The remote underwater camera. So this, I want to kind of talk about the importance of this camera specifically, because this really was a big part of actually using a camera for this project. There's a lot of people who have done underwater video kind of tests and different analysis, but there's a key difference in that theirs are all temporary, meaning they put it out that day, they leave it for a day, or they put it out for an hour and then move it, swapping memory cards, swapping batteries, all of that. This camera's out there and it's set. It's also a PTZ camera, meaning it can pan, tilt, and zoom, which other cameras also don't do, meaning we can really mimic it and make it look like an observer out there on the reef, kind of watching the reef. So this is what you would see if you aren't familiar with the camera. If you're watching, this is just part of the survey method, but it rotates, and then from here we can also go up and down with it and really get like a full perspective of the area. There really wasn't a blind spot by the end of the survey. We programmed it where we could get it to emulate a diver. And this also took many trials. It was me texting John late at night, try this one tomorrow, please, try this one tomorrow, please. Like, that didn't work yesterday. How can we get it? And what we settled on was it rotating where it makes a full rotation in about 5 minutes. Importance for that, again, you need to see the entire area in that species identification area. If you don't, then you can't say it's accurate. So we were working with that 5 minute period. But this is kind of what the reef looks like in the area. This is kind of the speed at which the cameras rotate, a little bit quicker in certain parts. Now to size and count with the camera, we had to get creative as well. So I came up with some scale bars we put out. So we had them positioned randomly throughout the area. We kind of placed some different depths and everything. We had the little 30-centimeter ones kind of placed over the bottom. And then we had what was supposed to be a meter stick turned out to be a yardstick because he got tired and bought the wrong one. But regardless, it was still there. We had a large scaling method as well. So using that, we had an idea with depth perception still, we could somewhat accurately try to size the fish. We also had to figure out how to mark the boundary. It's hard to tell what is, right, 15 meters when you're watching a camera. So we set out, it's hard to see, but there's these two floats. If you were there, you'd be able to see it. You know where it is. Hard to see in the pictures, but these floats were there marking that boundary as well for us. And then for this method, a single observer was responsible for the entire cylinder. So same as with the scuba. And for the sake of this, because this was a completely new method and I didn't want a lot of variation within this method itself, I conducted all of the camera surveys for the sake of being able to see if this one method was even consistent with one surveyor. From there, we did freedive surveys. So this one was really fun, but this was also what took a lot of time. We all became a lot better free divers as well, which is good. But really what we ended up settling on is 2 divers are responsible for the same cylinder. Many reasons, but the most important ones was just for safety. One up, one down. We were there with each other because we were diving all day. So you get pretty tired by the end of it. And then also from there, what we did is we alternated doing dives, and then we were each responsible for doing half of the cylinder. So we have a pipe, like you saw in the video, there's that pipe there. Basically, one would survey the north side of the pipe, one would survey the south side. So that was really nice to have. It gave us like a visual of where to stop your survey at versus not having that. But yeah, we'd both do half, and then we would be down there for 30 seconds each dive. So 30 seconds might not sound like a lot to some people, might sound like a lot to others. The reason we selected 30 seconds, though, is because it was what was feasible long-term, but also allowed you to accurately record species. Meaning we tried longer than 30 seconds. We did minute holds. We did 45 seconds. Problem you run into is being able to remember all the species you saw. Because if you haven't ever done it, or if you've ever tried, it's really hard to go down, hold your breath, and in 30 seconds, see all the fish and write all the fish in that same time. So what we found was go down, you observe, then you swim up to the surface and record the species you saw. This way we found it was much more accurate. If you're going for a minute, you forget many of the species you saw, and also you're just getting tired more so as the day goes. And then for counting and sizing, we did that from the surface. Reason being, holding your breath for 15 minutes multiple times a day is exhausting, just not really feasible. We want this to be feasible, right? We want people to be able to actually implement this. So what we did was we went down, did the ID there so that way we could see all the small fish, everything as well. But it also gives you that linear sight, so you have a better idea of how big the fish actually are. You're not just guessing from the surface, which is what snorkel will kind of be compared to. But yeah, this free dive survey, this was a lot of experiment, a lot of testing different methods to get to this point. Snorkel is exactly what it sounds like. It was the exact same as scuba, just doing it all from the surface. So this, again, now you can't really identify the cryptic species is what we expected. It's going to be harder to size things, but we wanted to test it and really see what happened. So we did that, and we found quite interesting results, to be honest. The first thing we wanted to do, though, was we wanted to make sure we were not biased in all the ways we were trying to avoid being biased. Meaning we wanted to make sure the only thing that was changing was the method and nothing else was at play. So the first thing we did was we ran a series of different Permanova tests. And these are really good just for looking at these different methods and compare them directly. So what this is just is that morning period from the afternoon. So all surveys included. And on the y-axis there, you're seeing the total species. So this is the mean number of species we were seeing in the morning versus the afternoon. As you can see, no significant difference and pretty similar. So that's good for us. We were avoiding those dial patterns. We were avoiding those schooling fish here and there. Next, we saw there's no bias in the survey order. This was important for us because again, we were randomizing it and we wanted to make sure that was actually being effective and we weren't kind of seeing, the first survey had more than the last. This also proved that the wait time we decided between surveys to allow for the disturbance in the settlement was enough where it wasn't significantly affecting it, because that was a big concern. As if you dive the same spot 8 times, are you now affecting the fish there or not? We found that you're not affecting them. So that was really important. Next, we looked at just a survey day. Again, like I said, it was over 9 days. The reason we did that, we didn't want any seasonal variability coming in. We didn't want the kind of the community out there changing on us, because as many people know, if you dive different times of year, the population of those assemblages. So we wanted to make sure that we weren't having that and we could avoid that. And again, no significant difference. So that concludes that, okay, we can focus on the method, and the method is really what's changing here. Between everything else, you can kind of, you can see is fine. So from there, now we looked at the individual methods. Are we seeing the same amount of species across methods? And this is kind of going back to that first objective, was can we see as much? And what I believed was going to happen was we would see more on the camera and less on the divers methods. What you see here, though, is actually quite the opposite. We actually saw the least on the camera and more on all the diver methods. And the reason for that, that I believe is what's happening there, is the camera, we program to do certain rotations, right? And at times you get stuck in a place where there's no fish. If you're a surveyor on the reef and you do that, you're just going to keep looking until you see fish, right? You're going to look for some. On the camera, sometimes we have that where you get stuck there. So that kind of can reduce the number. And then want to see, was this significantly different? There is significant difference across the total species richness. And really you can see here the lettering on each is kind of how you can group what one was significantly different than the other. So as you see, scuba and freediver both in A, those two are not significantly different versus scuba versus snorkel, or scuba versus RUV, excuse me, that is significantly different. So there's many different levels to this. And really what it comes down to when you look at it is that freedive and snorkel showed no significant difference. So no significant difference to scuba. And then RUV, the camera, showed significant difference to scuba. And the reason I'm honing on those is because, again, we're comparing them to scuba first. That's really what we're interested in. Yeah, there's some variance between them, but we can address that later. So overall, there is significant difference in the total species you're seeing. But as we go on further through this, you're going to see what might be able to explain that as well, though. So next thing we did was we looked at, we ran a series of linear mixed effects models. And essentially what this does is it's a model that's really good at taking into account fixed effects and random effects. And so in this, the fixed effect was the method, right? It was always the same method. We knew what we were doing. The random effect was the species of fish that was being implemented into the model. So as you can see in the formula there, the 1 bar species, that's telling it to kind of pick randomly the fish species for the model sizing. Now that's important also because this model takes into account that not all fish species are the same size. We don't want it to assume that every fish is the same size, so we should be getting the same results. We wanted to know that there could be variance in that size, and then how would it estimate it? So the bars you're seeing here, the graph, those are the estimate sizes. And then the yellow dot you're seeing, that is the projected or the predicted size that the model would predict that you would get. So you can see it's pretty close. The prediction is pretty close to the actual estimated size, but there is some significant difference here. So we did see that RUV and snorkel were significantly different than the camera. And the reason for that is different species being seen potentially. And this was what I was curious of was, okay, so RUV and snorkel, you're limited in what you can see. Snorkel, you're up at the top, you can't see the smaller ones. So, okay, wait, maybe we need to actually break this down and focus only on the species that were observed through all the methods, not include the ones that were only on some. So I removed all fish species that were observed less than 5% of the time. So this took the number of species down a little bit, but it did improve the model. So when you remove those rare species, the ones that only show up a couple times here and there, the prediction size did get a little bit better. And mostly if you see like the prediction close to that mean or that estimated value has gotten much closer. And that was huge because it was like, okay, these small ones might be throwing it off, are they? They most likely are. So we still had significant difference here though. And that was at the point where I was like, okay, so what is different here? What would still be throwing it off? And if you notice on the RUV, the camera method, the outliers extend much higher up. So that was for me, I was like, okay, what were we just bad sizing? Like, is that what was happening here? I looked back through the data and I went back and looked many times and realized there's a problem species in this whole thing by itself. And that is this guy. This is the big red lip parrotfish, right? The uhu. And these are very commonly targeted by divers. So what I think is happening here, and what I believe is happening, is on the camera, these uhu, they are not worried about the camera. They will swim right up the large one and come right in. Whereas when you're a diver out on the reef, these big uhu know what you are, and they're not going to come close to you. And this was kind of something where it was like, okay, but like if I remove it, will it help at all? And then you see in the model, it does. You see everything get much closer. There is still significant difference noted by the asterisk there, but you notice everything's much closer together now. And overall, like you see the predictions, a lot of them fall higher than that actual mean estimate. The reason for that, again, is you do have bigger fish that are just going to pull it up. A lot of the fish species are kind of in that 10 to 20 centimeter range, but you're going to have those bigger ones, such as like the uamamale, the orange spine unicorn fish, or the trumpet fish swimming through, things like that that come through that are much larger that will still throw off that sizing, which is kind of the outliers. But the majority of those fish are included there in that bulk. So that was really interesting to me. I thought that was really, really cool to see the fact that, okay, there is some significant difference in sizing here, but how significant is it? And is there just problem species? So from there, I wanted to also look at, well, what about sizing? Like, can we actually size these fish? And I kind of had my doubts. And to be honest, the doubts were kind of confirmed initially. Looking at this model, it's not a great figure, right? You see a lot of outliers, you see a lot of variance. But this has a lot to unpack as well. So again, it's the same model, same linear mixed effects. Now we're just looking at number of individuals versus size. And what you'll see, if you look at scuba, right, those outliers are going up to 100. I cut that off at 100. That goes up to like 300. And the reason is we have a lot of little Chromis Vanderbilti. If anybody's surveying, you've seen those guys. This area is full of them. However, where do you see them most? When you're down at scuba and you can actually see them and count them accurately. You can see in all the holes and everything. So the counting on scuba was significantly higher due to just that one species alone throwing it off, because on the camera, you can't see them very well from a distance is kind of what we realized. Free diving, again, you go down, you know they're there, so now you can count from the surface, but you still can't see them as clear as you did on the bottom. And then snorkel, we didn't include them at all because you couldn't confirm what species they were from the surface. You knew they were little damselfish, but there was no way to actually verify exactly what species. So again, I wanted to get rid of problem species like this. So I removed, the fish that were not observed more than 5% of the time. The significance. Obviously, right, it makes sense. These are all significantly different because that one species is pointed so off. And also, if you see like the predictor value there for scuba, it's not even within the range. Again, it's because like 90% of the count right now for that scuba was that one species alone completely throwing it off. So I wanted to take that out and see if it got better. And we found, yeah, that was a big part of it. And these schooling fish were mostly observed just on scuba. You're not seeing them as much on the other methods. However, you do see them, or you see now snorkel also is not significantly different anymore. So again, those little small fish can be problematic. And really make it difficult, but still on the camera and free dive say of things, there still seemed to be something going on. So I played around with this and tried to look through and remove problem species that I thought there would be. Really what it came down to was large schooling fish you can't avoid because they make up a majority of the fish out there, right? Like all the surgeonfish, the collays, the orange bars, all of these guys make up a major part of it. If I'm removing them from the model, I'm removing the majority of the fish. So really what it came down to is counting is going to be finicky and tricky. And there's a couple conclusions I came to. So overall, we should go with this, with the linear mixed effects models. Sizing works. Like you can size on all these methods, and even on the camera, we were able to size, and there was very little significant difference. We also found that the RUV is the best method if you want to look for larger vivorous fish, because it's the only one that picked them up. Again, we dove there many times, and that was the only time we had a parrotfish over 40 centimeters was with the RUV. The other ones were all under 25 centimeters. And that was consistent through all the sizing through methods. So knowing things like that, that's kind of critical, right? If we're talking about especially food fish. Counting, there was good takeaways from that counting. Yes, the models were ugly, but scuba and freedive are the best if you want to count cryptic species, which makes sense, right? You're diving down, you can actually see them, you actually know they're there, and you can positively ID them. And then scuba and snorkel are best for the schooling species. And reason is what I think is going on there is when you're up at the surface, you're not affected in schooling, and when you're doing scuba, they don't really worry about you. Free diving, you're coming up and down, you're kind of coming in quite invasive, but more so, a lot of those small schooling fish, things like cole, are also heavily targeted by spear fishermen. So potentially that's just a reaction to, they see spear fishermen, they go tuck and hide in holes. So there were good takeaways from both there. Overall though, sizing was a big surprise. It worked really well across all methods. So that was kind of how good are we at seeing what we're seeing? But at this point now, I want to know how good are the methods actually at what we're seeing? Meaning, are we seeing different species across these methods? Like we knew there was variance in there, and like in the models, right? I removed the ones that weren't present and all, so I got down to the ones that were there. But then I wanted to see, like, was there actually like strict divergence across this? So we did some ordination analysis, and we used what's called principal component analysis. Now, this method looks a little confusing, but it's actually a really great way to visualize very complex data. So if you see on the X and Y there, they actually have PC1, PC2 on the Y. So that's principal component. These are essentially made up of many variables that are changing within this. And when you set in this code, you get the results, you get numerous variables. But then you take the top two typically, and you plot them against each other. In this example, the components, or the principal components, are all the different combinations of fish species that were observed on a single dive. So we end up with a lot of components, obviously, because of that. It wasn't always the exact same dive to dive, but overall, these were the two most common and kind of the most effective towards modeling this. Now on here, if you look, you see all the dots, right? And each dot, you see different colors representing different methods. So the black being the scuba, the red being the camera, the green being snorkel, and purple being free dive. And there's small dots, and then if you look closely, you'll see a couple bigger dots. That's kind of where that median lies there. Or sorry, not the median. That's where like the overall like average of these points lies. And so this is all just presence absence. Again, this doesn't take into account counting or sizing. This was, did we see that species or not on this method? Did we see the species or not on that method? And what we found is, if you see, there's a lot of overlap, which is really surprising. I myself really thought there was going to be very clear separation across these methods. I thought, scuba would be here, freedive would be here, and camera would be up here. But overall, these methods have a lot of overlap. The one you see that's really kind of tailing off, though, is snorkel. So what's causing that? Well, if we look at the next figure that's kind of showing you what's pulling what in what direction, we can break that down. So again, if you're looking here, you see all these arrows, you see all the messy codes. I left those on there because I wanted you to see how similar everything actually is. There's a lot of overlapping, and there's not a whole lot of change across all these different species. So that's pretty significant, I thought. And then I want to see, okay, you see those three methods kind of pulling up that direction, and then snorkel pulling down to the bottom left. What is doing that? What's causing it? This shows us. So if you look, just focus on that top right corner of the figure, you will see those main species that have the strongest pull, the longest lines, are all small cryptic species. Why does that make sense? Because if you look back over there, that does not include the snorkel method, which we didn't expect to be able to see those. So that shows you, what are you missing on snorkel? You're going to miss cryptic species mostly, which kind of made sense. Other than that, though, all right, so why is the Abudev stuff down here? Why is it pulling bottom? Why is that pulling to the snorkel? Well, the reason for that is when we were diving there, you would see them off in the distance a lot, but they were kind of more surface dwellers. They were hanging out. They weren't really coming down to you. As soon as we go out there and we're on the surface, they either now swim up to you to see what you are, see what you're doing, or they dive down away from you and swim right to the transect. So there's some behavior there when you're at the surface now, because they're surface dwelling fish more so. And so that one was pulling in the snorkel. We didn't see that much in the other methods. We saw some, right? You can see there's some presence up in the diagram that overlaps. But overall, snorkel saw a lot more of the Abrupta. So really interesting thing here going on in terms of what species are pulling where. But in general, there's not much of effect of many of the species, and there's pretty clear grouping. So I broke this down a little further and just plotted like these problem species so you can see. And again, you can see separation, but again, not completely distinct. The reason for that is, right before I was saying, you're probably wondering, oh, I thought he said you can't count these, you can't see them, you can't size them. You couldn't do it effectively to an accurate number, but you still saw them sometimes, right? They were still there. We knew that, we just couldn't do it effectively. So that's why you still have some overlap. So this is the little one I was talking about. That's kind of the problem fish of the whole project. These little guys just kind of throwing things off. Not really a problem because they're just a part of it, right? But kind of are the species that tend to show variance. Again, another fish, you'll see it very similar to the last figure. This one is just the ornate wrasse though. Again, same thing. So the orange is when it's absent, the green is when it's present. So still overlap there, but generally you didn't see them on snorkel because you couldn't ID them. And then last thing, the belted wrasse. This one looks a little different. The reason being for this, there wasn't a lot of presence in general. This one was just not as commonly seen as the other wrasse. So smaller presence area, but when it was seen, it was on those top three methods still. So what does that mean? How can we summarize that? Overall, not much variance in these species. It's pretty similar, pretty equivalent, which was a big surprise. Overall, cryptic species are best observed on scuba, RUV, and freedive. So again, observe, not saying you can count and size on all those, just observe, you will see them. So meaning they're not going to change behavior. And then surface dwelling fishes are best observed on snorkel. And that's just because, again, they're going to kind of stay up or they'll just stay away from you. They're not going to come in. So all of that to me, I was like, wow, that's pretty interesting. All right, like definitely threw me for a loop, not what I expected. And then I really was looking at the camera and my thought was like, all right, I did this. The camera worked for me, but wait a second, like, am I being biased? Because I was out there diving too all day. So I'd go home at night and do the survey after diving the spot all day. So it's like, am I just assuming it's the same size as what I recorded earlier in that day? So I wanted to know this. I wanted to test this, make sure I wasn't just doing that. So I had three awesome kind of young scientists, people who have done some fish surveys, not a lot, wanted to test these methods. So what did we find? Found some pretty interesting stuff, which was good though. Overall species richness, total species we all saw was pretty equivalent. You can see a little bit of variance there, but overall nothing crazy. Most likely, again, the small cryptic ones hiding in the back, just not seen. But overall, the results remained consistent, this was huge because for me, I was really nervous. I was like, oh, there's no, like, I was just assuming it was the same size. I knew what it was or whatever, but I wanted to know. And they kind of confirmed the sizing is pretty consistent. We're pretty good at sizing on the camera, counting, still a lot of variance, just like I had. So yes, it's not a perfect method, but the method itself is consistent across surveyors. And so we can build on that from here and kind of see what is the next step. What do we need to change about it? And then from there, I was like, oh, now I have kind of some data that's interesting to compare to old kind of conclusions people came to. And one was that there's these problematic fish families on the reef that new divers aren't going to count and aren't going to ID the best and kind of, yeah, just do accurately. So I was like, okay, well, I guess I have that now. I can compare that. So we did that as well. And we looked at, this is the sizing for these problematic families. So Acanthuridae, things like the surgeonfish up there, you see the sizing pretty similar. Surveyor 1, this was actually kind of consistent across all fish. Surveyor 1 was a little over what I put as what I had as the mean value, right? Surveyor 2 and 3 were kind of back and forth on right there as well. So this kind of showed the method is pretty consistent because it was just these surveyors had the same consistent result compared to what I had. Pomus ventridae, though, if you look at that, you're like, what? That's totally wrong. There's no way what you're saying is true. This is actually, again, that Abrupta species. You remember me talking about the one that was up at the surface, hanging out on the ridge. What's likely happening here is Surveyor 1 saw them and they considered it within that cylinder, that 15-meter cylinder, where the other three of us, it was like, nope, still on the edge, not going to count them. And then that drove that fish family average way up, because those are like twice the size of the little chromis, which make up majority of those. So we have that. And then counting, again, we'll just kind of continue with this. It wasn't great. The counting was fairly good though for like the major fish. Sorry, it says size there, but these are the counts. So it was fairly consistent across it. But again, sizing still needs to be improved. So going back to what I said, what I started with, what did we really find from this? That's a lot of stats. That's a lot of results. But what did we find? Again, we were trying to enhance our ability using camera, using freedive, using snorkel. Again, I thought that the RUV was going to be different. What did we find? Diver-based methods did not affect fish behavior, and they did not result in lower species in RUV.
Let's turn to meadows.
That was kind of exciting. And then on top of that, the limited field of view in the RUV was what resulted in it. So we were able to figure out what it was causing it and what do we need to change moving forward if we want to get more accurate. And in all methods are complementary, which is huge. I mean, being able to use these other methods in these areas can really push the boundary of what we're doing and also the inclusion of who's doing it. From there, again, objective 2, what was it? We wanted to examine the relatedness using these different methods or using the different analysis, and expected there to be a lot of change. What did we find? Well, all methods proved to be capable of sizing. So we could say that was a win. That was better than what I thought was going to happen. And then if we want to talk about counting, you really got to look at what species are you focused on. If you want to count cryptic species, you better use scuba or freedive. If you're doing snorkel, you're going to miss a bunch. The camera, again, you'll see what cryptic species are out there, but you're not going to be able to count them effectively. And then for schooling fishes, as well as scuba and snorkel, those are going to be your best methods. Because again, we saw that significant difference change. And basically the schooling fish, the schooling species, for whatever reason, were more present on those certain methods. So once again, all methods are complementary in some way. You just got to figure out which way you want to use each method. So from there, what is really the future? What do I think is the best thing to do moving forward? One, I think we need to utilize the technological advancements to enhance the effort. So yes, there are some things it can't do, but there's a lot that it can do. And this is kind of that first evidence that these cameras, these remote cameras, can actually serve ecological purpose. They're not just there just for viewing for fun. And then second, increased collaboration between researchers and communities. And I think this is just as important because we can say, look, we have advancements, we can continue to go that direction, but also we don't need all this gear, we don't need all this technology. You can grab a snorkel and a slate and you can go do the survey too, which is also pretty big because there's always been this separation of you have surveyors and you have scientists and then you have fishermen, and they don't really all work together. They don't get to do these activities together and kind of figure out what's out there together. And this is a way where we can get many more in there. And then also, again, I'm saying these are complementary methods. These are not to replace scuba diving. This is not saying scuba is wrong and is inaccurate. No, this is, okay, if you have other things you're interested in, this is what you want to use. And these are the methods that you can use. So continue those long-term studies we've been doing. Don't end that. And more specifically, more cameras. That's what I think. I like cameras. I think they're great. But really, cameras like this proved these can be like true ocean observation stations. So when this started, the goal was the company that funded like the first camera, their goal was to have these stations looking at oceanographic changes, so temperatures and things like that. Now with the camera, we can visually see as these things change, biologically and ecologically, are we seeing changes matching all of those oceanographic features as well? I think that's huge. We can also provide data about the dial patterns. We got year-long data now. Right now, surveys happen certain times of year when the weather's good, when it works for everybody. When the camera's out there now, you can use whatever day you need. So that's great also. And then lastly, two is better than one, meaning scuba and freedive, they don't need to be separate. Honestly, if you think back and you looked at what was different here and there, if you put scuba and snorkel and freedive together, they're pretty much the same as scuba. So this is great because now this means you don't have to be an experienced freediver to be able to effectively survey. You also, if you're a snorkeler, you can. So there's pros and cons to each, and we kind of get to blend the groups now together and really bring in kind of the community aspect of it. And you don't need to just separate them. I think the 30 seconds that we did is great. It's a great way to go about it. But honestly, I don't think that's like the key thing about it. I think more so you just need to dive down and find the cryptic species and know that they're there and confirm the ID, and then you can just go back and count. If you dove down 3 times versus 5, you'd probably get pretty similar results as long as you continue that surveying from the surface as a snorkeler. So that was really, really cool. And this was an exciting, honestly, finding for me. So that being said, a lot of thanks to you out for this project. I had very little to do with actually what got it into place and got the pieces there. I was just able to use a lot of resources to my advantage and conduct a really study that I was hopeful that could work, like John was saying, and thankfully did. So Omega Lab, first off, I've never worked in a lab even close to like the Mega Lab. Honestly, like the group of people you're surrounded with, the way everybody like pushes you. And honestly, the coolest part for me was I came to John with a lot of ideas. And the one thing I never heard was no. He literally just said, go try it and then tell me what happens. And like having that freedom as a scientist, like that's a pretty cool like place to be, because if you get told no all the time, you kind of just want to like stop and just want to move on, right? So for me, that was a big part. Fathom, they're the people, the brains, the engineers John was talking about behind the camera. So spending the night with Trevor, learned a lot in that one night with him. That was great. Change Hawaii as well for some funding with the project. Aqualink there, again, they're the ones who funded that first camera as well, got it going. And then TCBES and the dive program was great. And then more specifically, my committee, Dr. John Burns, Dr. Randy Stuckey, and Dr. Grady up here up front. These 3, honestly, like they gave me the reins to do it. They never held me back. They didn't give me answers either, but they really pushed me to find the answers. And I think we did, which is really exciting for me. Honestly, it was definitely an honor for me to work with the 3 of you and get this project done. Divers, huge thanks to them. Jacob, Wesley up here front, he didn't miss a single survey day. He was one of the rocks of getting the actual project finished, because there's no way I could have done this alone. So that was huge. Also helping people get in and out all the time. So that was grateful. But also Kylie coming over from Oahu, she came over for a week, helped us out. Kukui, Ashlyn, you guys filled in whatever needed. You were never complaining. You just showed up when you could. That was amazing for me. Then video analyzers, Makoa, Manuel, and Erilyn, thank you for doing these things last minute when I come with a crazy idea at the end of my project. My buddy Chris for doing freedive training for us. And then also, like I said, that year of pilot data, I asked a lot of questions. So I asked Atsuko, I asked Kylie, Crispin, kind of all these people, all these questions, and they always help the entire Mega Lab. So thank you to everybody with that. That being said, any questions?